Welcome everyone to our webinar today. My name is Travis Avery and I am our Regional Agronomic Marketing Manager with Taurus for Central and Eastern Manitoba. And today I have a special guest here to talk about mycorrhizal fungi and Rhizobium Bacteria. And the guest is Joseph Gardiner from Joseph Gardiner Farms.
Welcome. Hey Trav, thanks for having me.
Cool. Well, let's just jump right into it here and get to know your farm a little bit better.
Cool. Yeah, so like Trav said, I am, my name's Joe Gardiner. I am the owner, operator, manager of Joseph Gardiner Farms. I farm with my parents. We manage 19 quarters, 1,600 arable acres of land. We got 250 head of beef cow right in the middle of calving right now. Yeah, our farm's always been, I guess, somewhat open to new ideas. My parents in 2002 did the holistic beef management course, so we've kind of done some different techniques as far as managing our beef herd. As far as myself personally, I'm a soil healther, I guess. I've been researching it for the better part of going on a decade or so. My first cover crop was in 2013. We did lots of trials, lots of different stuff. That was a time when I was going to a lot of conferences, doing a lot of reading, podcasts, documentaries and stuff, and kind of had to figure there was a better way to do things. So our farm went full regenerative in 2018. What does that mean? There's no one set definition for regenerative agriculture, but we basically intercrop everything. So unless it's a perennial grass that's growing that we're growing for seed, we intercrop everything with different species. We incorporate the cattle as much as possible. We've been zero till for about 15 years. And yeah, trying to reduce herbicides and trying to use fertilizers more efficiently. As far as cover crops, basically the theme we go on is we try and just match different warm and cool season cereals with warm and cool season legumes, sprinkle in some brassicas, just try and mimic nature and create as much diversity on the land as possible. So that gives you a little background of kind of where I come from and what we're doing here. Trav, if you got any other questions?
Right on. Well, I do have a lot of questions, Joe. That's kind of how we're going to structure this little webinar, but basically just going to pop up a screen like this. We've kind of went through this, you and I, before, so we just want to, touch on certain questions. This one specifically, who are your influencers on your path to, building a healthier soil on your farm?
So I was really lucky when I went to the University of Manitoba, and I met a professor by the name of Martin Entz. He was the organics professor at the U of M. But I never have had a huge urge to go organic, but he really presented a different way of farming, that farming didn't have to be input. We can actually use management skills and different crops and livestock to get the same desired result, which is profitability, but a different way of doing it and utilizing the resources you have on your farm much better. So Martin, I have to give credit to. I wouldn't be here. I'm sure I wouldn't be farming today if it wasn't for him. He got me excited about the idea, and it's kind of kept my enthusiasm right through. And today I'm more excited for the future than ever before.
Very good.
I would suggest all of the people on the screen right now, every one of them, kind of the uniqueness of them, or the difference between them is their uniqueness. There's scientists, there's farmers, there's entomologists, there's university professors, and people that didn't graduate high school, but they're all amazing people with different stories, and kind of in this soil health world, we can't learn it all, we can't try it all, so we need to kind of come together as a larger group, a larger peer group, to share each other's ideas and our trials, and both positive and negative, so we can further benefit one another.
Yeah, very cool. Definitely goes to show the systems approach that you need to adopt some of these soil health principles. I noticed you have everything from a first generation farmer to a microbiologist on this list, right? So it is quite the journey. That's an understatement a little bit. So anything else?
Couldn't agree more, Chav.
Pardon?
Couldn't agree more.
Yeah, perfect. All right, well, let's dive into something a little bit different here. But I typically like to show this presentation and ask farmers, ask growers, what's the difference between these two pictures? There's 450 million years of research and development is the difference. So, Joe, can you tell me what the picture is on the left?
Yeah, isn't it Mars?
It is. It is a picture of Mars, and the picture on the right is a picture of a native grassland. So I think what we want to drill home on this point is just the fact that we got to start paying attention to some of these biological processes in the soil, some of the stuff that nature's taught us for many years. So, and that's kind of the whole idea around this webinar is to actually dive into some of these biological processes and how we can start to harness nature to grow some better crops. So.
So Trav, you're a lot more in tune with the chemistry side of it, and you've kind of been my soil test guy for a long time. So I'm fortunate to have you that I don't necessarily have to know the chemistry stuff because I don't find it as interesting as the biological side. But correct me if I'm wrong. If you took a soil test of Mars there, it might look okay on a soil test.
Yeah, actually, NASA proved that. NASA actually had a report out to say that they could take every essential element off of Mars and that a plant requires to grow. Obviously, the difference is life. That is what Mars doesn't have, right? So, yeah, it's just goes to show. Like, let's really dial into these biological processes because we sometimes I think take them for granted. So, that being said, I'm going to hop into this slide. Arguably, actually undisputed, the number one most important biological process would be photosynthesis. Somehow plants have figured out a way to convert sunlight into sugar and oxygen. That oxygen is for us to breathe, and that sugar is to build the plant, which is, when you really think about it, something really astonishing. So, anything to add on this slide, Joe? I know photosynthesis is true to your heart.
You hit on most of the good points. Without photosynthesis, it's a non-starter. So we need to find better ways to do it more efficiently and, most importantly, more often.
Yeah, for sure. I like to say it's our jobs as farmers and agronomists to manage what we have in the yellow here, manage mineral nutrition so we can maximize photosynthesis and minimize stress, because this glucose molecule is the building block to everything we see here. So,
and to touch on that, Trav, from a farmer, our job is to give the means to be able to do that. And what does that require? A growing plant, a seed in the ground that can capture sunlight.
Yeah, very well said. So, yeah, we're going to pay attention to one thing on this diagram. This carbon has root exudates. There's lots of stuff we could, but that carbon has root exudates. We're looking at a picture of a root exudate coming off a plant right now. But I typically like to ask why. Even when I saw this picture for the first time, I always thought, why is a plant giving up this a third of its carbon to the soil, right? So, but actually, those root exudates are the communication system between the soil and the plant. If we take a minute to really think about that, it truly is astonishing that these plants and soil have evolved together from the start of time. So. Any comments on root exudates, Joe? I know it's a hot topic these days, but
you touched on all the good points, Trav. I think what I like to think is a plant doesn't have the ability to build itself. It takes that carbon, it gives it as an offering and instructions to the biology, and that biology goes in and gathers the minerals, brings them back to the plant, and literally builds that plant. It doesn't happen without those root exudates.
Yeah, quite crazy actually when you really dive into it and think about it. So, but what we're gonna do today, we're gonna have to speed this up a little bit. But anyway, we're looking at two soil microbes. We're looking at Rhizobium Bacteria and Mycorrhizal Fungi. So what we're gonna do, we're gonna dive right into Rhizobium Bacteria and the symbiotic relationship it hosts with the plant, and ask Joe about the importance of Rhizobium Bacteria to your farm. Why are you utilizing Rhizobium Bacteria on your farm?
Yeah, so the reason, one is we are trying to mimic natural systems, but two is I'm trying to lower my operating load as well. So why, when there's so much nitrogen in the air already, and these little bugs that you see on your screen right now are willing to go out, gather it from the air in the soil pores, and give it to the plant for free, it just, from a regenerative farming standpoint, one, it makes sense for the soil, but two, it makes sense. I'm not cutting a nitrogen check, a substantial nitrogen check. I still am, but yeah, it's something we can focus on, and we can utilize rhizobia to lower our synthetic nitrogen needs.
Yeah. I would say, how many people would know that 78% of the atmosphere is actually nitrogen gas?
Trav, I know we're short for time, but Gabe Brown has a classic line. If you take a 2,000 bushel bin and you fill it right full of 4600, there's more nitrogen in the atmosphere above that bin than what's sitting as a granular form in the bin.
Interesting. Yeah. So how do we utilize that nitrogen above the bin, right? So there's two ways we're doing it right now. We're doing it through the Haber-Bosch process, and then we're doing it through soil biology. So I look at soil biology as like you said, an easy way to reduce the operating and utilize what's in nature. So and just to touch on this a little bit, like if this is your simple uptake and removal chart, but if you're planning on growing, even if it's 50 bushel soybeans, you're going to need 217 pounds of nitrogen to grow those 50 bushels. So that's pretty self-explanatory as to why we should be inoculating our legumes, our pulses with Rhizobium bacteria. So yeah, fascinating stuff from a Rhizobium bacteria front. So anything else on Rhizobium for now, Joe? We're going to dive into mycorrhizae here right away.
Well, and we're going to talk more about soil pores, but you can see, Trav, like 217 pounds is a lot of nitrogen. If we don't have pore space for that air and that nitrogen to come in via pore space, then that Rhizobium bacteria has to pull it from our organic matter or from a less sustainable source inside the soil profile, not air. So if we have less air in the soil, we have less means to break down that nitrogen, and the rhizobia and the plant signaling to the rhizobia, I need more and more and more. So it's got to get it from somewhere.
Yeah. I think you're segueing for a little bit later in the presentation, but yeah, great thought on pore space and the importance of soil aggregation. So we'll touch on that in a bit. Let's dive into mycorrhizal symbiosis. So before we do that, we're just going to play a quick video. This was when I first came on with Taurus. This was a video that really helped me understand mycorrhizal fungi. It's basic, it's simple, but it does give the overarching understanding of what mycorrhizae is. So,
how does this symbiosis take place? Mycorrhiza is the term used to describe the symbiotic relationship between mycorrhizal fungi and the plant's root system. Through that symbiosis, the fungi and the plant naturally develop and mutually reinforce each other without harming one another. Here are the 3 steps that explain how the symbiosis between mycorrhizal fungi and a plant take place. Mycorrhizal fungi initially germinate in the soil and make their way to the nearest roots. The roots are then colonized by the fungi, and the mycorrhizae are established. The fungi penetrate the root and create an internal network of fungal structures where the plant and mycorrhizae exchange sugars and nutrients. Finally, the hyphae continues to develop outside the roots to explore the soil. Basically, mycorrhizae form a network of fine filaments that associate with the plant roots to draw nutrients and water from the soil that the root system would not be able to access otherwise.
What do you think, Joe?
That is a pretty cool video, Trav, I think.
Yeah, I liked it too. So
it breaks it down, it makes it pretty simple. When you start talking about fungi and species and distances and absorption and chemistry, it can get very confusing, but that video breaks it down pretty simple.
Yeah, I thought the same thing. So here's another way to break it down pretty simple from this picture, but we're looking at a root, and then we're looking at root hairs here. Obviously, we all know that plants absorb nutrients through root hairs. I think the cool part about mycorrhizal fungi is this hyphae that extends outside of that root hair system. So if we can promote this hyphae network, we're going to be better off for mobilizing nutrients and water in times of need. So
and to touch on that, like at the bottom you have hyphae absorption 10 times.
Yeah.
Like there's oh, I can't pick out a specific paper, but there's lots of research out there that absorption can be way greater than 10 times. It could be 100 times or even 1,000 times in some diverse species mixes. But if you can add that power to your root system, why aren't we focusing on this more?
Yeah, I would agree. It is it's a powerful little fungus. So this picture actually makes it seem a little bit more powerful because we can actually, picture what's going on in these hyphae. So this is actually an exchange of nutrients for carbon going on in the hyphae. So
Trav, that is a 4-lane highway, and they're all semi trucks packed with nutrients.
Yeah, I like great analogy. Love it. But yeah, great little microscopic picture on mycorrhizal fungi. So, but yeah, there's your basic understanding of mycorrhizal fungi. I'm going to ask Joe the next question here. What was your aha moment when it came to mycorrhizal fungi?
Yeah, so like I said, or I touched on at the start, Trav, we started intercropping, and the one I wanted to go through here today was corn vetch, because obviously corn is a very popular crop, but there's just little things we can do with our just little ideas to incorporate diversity that can make such a big difference on all the things that we were talking about today. Rhizobia, mycorrhizal fungi, soil aggregates. So this corn vetch intercrop I did in 2018, and kind of the idea is this is the start of it. But the idea is we're just using the vetch to gather wasted sunlight that was hitting the soil surface, and because vetch it's a legume and a very, very high nitrogen producer, as it stays in vegetative because it doesn't have much sunlight to hit reproduction. It is just pumping nitrogen. Those rhizobia are pumping nitrogen into the soil profile, and then we're using mycorrhizal fungi to transfer those nutrients, nitrogen to the corn, and obviously the corn is giving back minerals that the vetch is in need of as well. So it's a pretty cool combination. We do it. It's a regular practice on our farm now. And there's lots of trials going on. So it's pretty exciting stuff for the future. But we'll go through it here. As you can see, we sprayed this with Roundup. The vetch is somewhat resistant to the Roundup, but the corn has a chance to get a head start.
Hey Joe, sorry to interrupt you, but to go back just to how you seeded it, was the vetch first? Was the corn first? How did you seed the
You bet. So we sowed the vetch. We put down phos, just water-soluble phos, which I think we might touch on later, was a big mistake when we're talking mycorrhizal fungi. And then we put down, it was lots of nitrogen, 150 pounds or something the fall before. So the vetch was sown, and then the corn was sown the following day with a 30-inch planter.
Okay, cool. All right.
So you can see when these plants start to hit symbiosis. So the corn's getting up high enough, and the neat thing is the vetch is a cool season, so the vetch doesn't like full sunlight. So you can see the difference in 5 days from July 6th to July 11th. The corn has the ground properly covered, and now it just made conditions proper for the vetch to thrive. So we can go to the next.
Your next picture is the coolest here.
Yeah, you betcha. So you can see just a few days later how thick the vetch gets. By the end of July, like if you remember at the start of July, there was basically no growth. By the end, the corn's 6 feet tall and there's a mat of vetch. And just what I want to touch on is you can see the corn's getting all the sunlight it needs, but there's still wasted stuff down the rows. So up and down the rows, there's little blobs of light. That's what that vetch is living off of. It is just wasted energy from what the corn leaves were missing. So kind of neat that we can take advantage of all that energy on all facets and get it into the ground where it belongs.
Yeah, we talked lots about maximizing photosynthesis. Boy, are you doing it here. Very
For sure. There is, and just a quick story, Trav. I took one of those laser thermometers out, moved the vetch aside. This was on like a 30-degree day, and took a temperature, and it was 70 Fahrenheit, which is, if you when we're talking biology, that is the ideal temperature. I walked to the edge of the field on a black spot that had direct sun, and it was 100 Fahrenheit, which means it was so hot in the top centimeter that biology had shut down. It was too hot for them to work in those conditions. So under these conditions, we have totally regulated ideal temperatures for biology. Where we have black dirt, it had shut down because of heat stress.
Fascinating stuff, Joe. Yeah, well, I'll pop into this. This is one of those things. I thought we'd throw this in here just because, we've been taught, or I've been taught my whole life that, those plants in between the rows would be considered competition. We actually know that to be the opposite in this case, where, this was one study showing the nitrogen transfer between legumes and grasses, but it summarized it in 3 ways. It's a direct transfer of nitrogen through the mycorrhizal hyphate network. The other way that it happens is the decay of plant tissue, so that legume dies and then decays, and then the exudation by living cells. So there is an incredible amount of research papers in this field of study being done now, understanding intercropping, understanding which species are working best together. Exciting times, anyway, to better utilize things like Rhizobium Bacteria and Mycorrhizal Fungi and the whole soil food network down there. So.
And Trav, the exciting thing, mycorrhizal fungi in agriculture, it hadn't been studied hardly at all, even 10 or 15 years ago. Yeah. And in the last 5 years, there's been some amazing studies, but the exciting part is there's lots we don't even know about this fungi.
Yeah. One of your influencers talks about that quite a bit, Chris Nichols, how much she's learned and how much she doesn't still know, even though she is a soil biologist. So.
A brilliant soil biologist, and she will admit she doesn't know 95% of what's going on in the soil.
Yeah, fascinating stuff. So, but anyway, just to finish off this corn veg, Joe, here's September. Not much to, well, obviously you could talk about this all day. Yeah. But let's
basically
into this water infiltration here, Joe.
You betcha. So this is kind of the eye-opening, like you talked about, Trav. We create soil aggregates, we create pore space. And when we're talking water infiltration, that is exactly what we're talking about. Is basically, yes, it's how fast the water goes in, but it's telling us directly how much soil aggregates we have and how healthy the soil aggregates we are. So I did a test on similar ground with corn silage, and we did 1 inch in 55 minutes, the second inch in about 12 hours. Obviously, I wasn't there when it ran out. But the corn vetch, this is impressive, even from a soil health guy. You won't top these numbers, which is 1 inch in 2 minutes, 2 inches in 22 minutes. Even on my full season cover, which I hope we have time to talk about at the end, we don't do numbers that good. And basically just what we did is we balanced the soil, giving perfect segue. I wanted to talk about carbon and nitrogen. So basically, I just want you to focus on the ideal microbial diet is 24 parts carbon to 1 part nitrogen. So you look at corn stover is 57 to 1, and you look at hairy vetch cover crop, which is basically what was there, is 11 to 1. So the only thing we have done is given the microbes a balanced diet. So we've given them high carbon corn stalks and stover, and super high nitrogen, low carbon, very vegetative hairy vetch. And what happens is exactly what we talked about there with the water infiltration. We just have stronger soil aggregates because of biology and because of mycorrhizal fungi transferring those nutrients. And as you can see from the photo, there was just an absolute slew of earthworms. I was pulling shovelfuls that was getting 10 to 15 earthworms per shovelful. It was just it was really amazing, but it was all possible because we balanced this carbon to nitrogen ratio. And a benefit of that as well is our residue will be cycled quicker, and those minerals will become made available faster if they're balanced. Right? Everyone knows how long it takes for a corn crop, if it had to decompose on its own, how long it would take. But if you mix a low carbon, high nitrogen, you blend the diet for the microbes, and they'll cycle those nutrients quicker.
Yeah, I think that's a great point in understanding how to and when to utilize cover crops. A lot of folks might say they tried cover crops and didn't factor in the carbon to nitrogen ratio, and then end up saying they didn't work, right? So I think this is a conversation for another day, understanding this thing completely, this carbon to nitrogen ratio. But boy, is it important. And
Yeah,
look what you
Trav,
found.
I would highly recommend, if people were watching and they haven't heard of carbon to nitrogen, obviously look into it. But even if you just take a picture of that graph, all you need to know is, the ideal diet's 24-1. That's going to break down relatively quickly. Like alfalfa residue hardly sticks around at all. And I mean, that promotes a living soil. So if we can focus more on that number, we ensure that our biology has a viable food stuff. They're not pulling out of our minerals in the soil out of our organic matter, and they're creating that precious pore space. So we have a functioning soil. So yeah, it's not that difficult. If you take a picture of that on your phone, and just when you're thinking of rotations, just be mindful of that. The faster decomposition you have, not all the time, but if you balance it at 24-1, you're going to have a healthy living soil.
Yeah, very, very good advice, Joe. That's something I wish I would have learned a long time ago. Cool
Same,
stuff.
Trav.
Anyway, first take of time, we got to move on here, Joe. But maybe another series, get you back on talking carbon nitrogen. I think it's such an important piece to this whole soil health movement. So,
I think so too.
Is key on that. But anyway, sorry to kick you out of carbon and nitrogen, but we're going to move on, and we're going to ask you a question here about when you're actually selecting a crop for your rotation, do you factor in its relationship with mycorrhizal fungi? And if you do, why?
Yeah, so I would say this is actually more important than carbon and nitrogen. Is we need to focus on crops that keep fungi, keep a symbiotic relationship with fungi. So for example, perfect segue, Trav, we don't grow monocrop canola on this farm because brassicas do not form that relationship. And we see it. We see these canola crops being more susceptible to bugs, more susceptible to airborne diseases, more susceptible to soil-borne diseases. Like it's not rocket science. Mother Nature figured this out a long, long time ago. 450 million years, fungus created symbiotic relationships everywhere in the world. And now we are just growing large pockets where they don't exist at all. It is an information center. It would be like dealing with COVID-19 without the internet. Like it's devastating. To be honest, Trav, we still grow canola. We grow peola. So we'll grow specialty peas and with canola, more for the purpose of holding the crop up so the peas don't get diseased. But yes, to answer your question, we have plants on every acre that form symbiotic relationship with mycorrhizal fungi because they're so important for soil life, soil development, and I'm gonna hit on it again, but soil aggregates.
Right.
So the list that's up there, it just talks about plants that are dependent on it, somewhat dependent, and then brassicas do not form a relationship with mycorrhizal fungi. They have other important things in nature. I'm not crapping on all brassicas, but they evolved with mycorrhizal fungi as well, just in the form of other plants having the relationship with it.
Great. Yeah. Yeah, I like this. I like this chart too. I think a lot of folks can resonate with the dependency chart here. When we look at corn, we look at flax. Flax being one for sure for any of the guys that grew flax years ago. But just the yield penalty essentially you'd be taking on growing flax on canola ground or corn on canola ground, which we still see quite a bit still today. So definitely, I know the chart's blurry, but definitely it gives a good depiction of what's going on here with mycorrhizal dependency. So
for sure.
Joe, I like to tell a story about phosphorus deficiency in corn. But I was actually at a colony last year. We were talking phosphorus. We were talking crystal green predominantly, and he wasn't that interested in crystal green, mostly because he had 20, 30, 40 parts per million phosphorus in a lot of his manure ground. So anyway, he kept me entertained, to say the least, for a while, and we got to this slide. It really hit home for him because he looked at me and he said, "Well, I actually saw that in my corn last year. What the heck's going on, Trav?" So we got the next slide to follow that up. Phosphorus agronomy 101, but when we put phosphorus in our soil, or if we have phosphorus in our soil, it's not moving. That's where it's staying. So we couple that with corn. Here's a phosphorus uptake chart of corn, right from V3 to R6. But we notice how much demand corn has late. Not just corn. Every plant that's photosynthesizing, it requires phosphorus late. So to summarize that, we looked at this picture, looking at our root and looking at root hairs, and then we looked at this depletion zone. So when that plant is demanding phosphorus late, and say typically we've depleted what's in this depletion zone, or what's in the root hair zone, how is that plant taking up phosphorus when it needs it the most, right? If it doesn't have this you can see this mycorrhizal fungi network here. If it doesn't have that association, that plant ends up being phosphorus deficient. So we dug into it a little bit more, and sure enough, we found that this guy had grown his corn on canola ground. Told the story to me, and told the story to himself. So it was an aha moment for both of us.
Well, Trav, why don't you take a minute and you were the one that has educated me on phosphorus, but take a minute to talk about water soluble phos and mycorrhizal fungi.
Yeah, well, actually, when we apply water soluble phosphorus to a root, it's the same philosophy actually as Rhizobium bacteria with nitrogen. We know when we put down lots of nitrogen with our soybeans or peas, it will deter Rhizobium bacteria. It will create what is called, I guess, lazy roots. The same can be said for water soluble phosphorus and mycorrhizal fungi. If we put a lot of water soluble phosphorus in that rhizosphere, it's going to deter mycorrhizal fungi from forming a relationship. That plant is not going to want to give up carbon if it has phosphorus readily available, right? So I think that's one thing we should pay attention to as well. How can we start to mimic this natural system using things like water insoluble phosphorus, or using things like mycorrhizal fungi to do so. So does that answer a little bit, Joe?
Nailed it.
Yeah, definitely a different way to look at it. But if we really want to utilize mycorrhizal fungi, that's a great start, I think. So sorry, Joe?
Couldn't agree more.
Yeah. I'm going to pull it back to research here for a second. This was actually a trial. So again, Premier Tech does quite a bit of third-party research all over Western Canada, but this one stuck out to me because it was up in Swan River. It was soybeans growing on canola ground. But what we actually saw up against all the leading competitors, and the leading competitors were just straight Rhizobium bacterias. But we actually ended up finding, between 7 to 8 bushel yield difference. We don't see that consistently, but in this case, it told the story, right? The fact that we grew soybeans on canola ground, and we found, consistently on a replicated basis, a nice response. So definitely, it basically just wanted to tell the story around mycorrhizal fungi and canola ground. So
well, and soybeans are dependent.
Yes. Back up to that chart. Yeah, they are dependent species on mycorrhizal fungi. So same can be said for peas. Same can be said for other legumes. So very cool stuff. Let's switch gears here, Joe. We're going to switch to water. Tell us about water and why mycorrhizal fungi is important to water uptake in your farm. Maybe not just uptake, but everything from infiltration, holding capacity, efficiencies, runoff.
Yeah. Well, it's gonna sound like I'm beating a dead horse, but soil structure is so important. Mycorrhizal fungi, I'm not sure if we have a picture in the slide or in the show or not, Trav, but mycorrhizal fungi just wraps around soil aggregates like a net. And sorry, I just saw you had that other slide up, Trav. I was going to touch on that for a minute. But wraps around it like a net, making these very powerful, very stable root exudates. So we have a room for functional biology, room for water infiltration. But this study is amazing. I read it, I don't know, a year or so back, but it's a university study. I forget where, but it talks mycorrhizal fungi having the ability to store water for later use. So it can not only transfer from one plant to the other based on who needs it the most, it can store it there as a water rainy day fund. So like we talked about earlier, like there really hasn't been, when you think how complex fungi is, there hasn't been a lot of research on it. And like, we're just going to find out more and more and more cool stuff just like this about mycorrhizal fungi.
Yeah. Yeah, that is such a fascinating picture on the right there, Joe, seeing the water actually inside the hyphae, like you said,
Yeah.
the rainy day fund. Boy, is that fascinating stuff. So
Yeah.
excited to see the research going forward from, academia and universities. It's exciting times in agriculture for sure.
And on farm.
Yeah. Yeah, absolutely. How can we start to tell your stories a little bit better, right? So
For sure.
we're gonna
Not
start
just
with-
mine, lots and lots of farms out there.
Yeah. I just to paint that picture a little bit better. I saw this picture from Green Cover Seed, a presentation of Green Cover Seed, but what they're showing here is actually the 2012 drought in the States. So obviously there's a cornfield in the background, and then just native prairie grass in the front. But as you see, maybe the mycorrhizal network is in full function here in the forefront in the corn. Maybe there wasn't the mycorrhizal network, but the next picture is actually what's really fascinating. So Joe, what do you see here? Can you tell me what's going on?
Oh yeah, totally. So the green patch is balanced by nature. There will be a blend of cereals, legumes, forbs, and most importantly, a very dense fungal network. And they're working together. You don't see certain bulb patches. It's lush, it's green. And then obviously look over into the corn. Why are those corn stalks green? Well, they're on the edge of the field. They're mycorrhizal dependent. They've tapped into the native system, and we're able to extract water, able to extract minerals, whereas 10 feet into the corn, the network doesn't go that far. And the corn is super drought stressed, we'll say at the very least.
Absolutely astonishing. I absolutely love that. Thanks, Green Cover Seed.
Yeah, totally. It's a great picture.
And I'm going to show one more trial here. This was actually done in Portage at ICMS, replicated trial. Again, it was on canola ground. But what we found, again, about a 4 bushel increase across the board with Rhizobium mycorrhizae. But the story to be told here comes with rainfall. So, they documented rainfall throughout the year, May, June, July, August. But look what we find in August. We don't see the same rainfall in August, which is typical for us in Manitoba when we're growing soybeans, right? Typical across Western Canada for August to be a little bit drier. So, I think this correlates perfectly to the fact that we saw maybe that 4 bushel increase. Maybe that was the reason why, because we had that mycorrhizal network, pulling in water that those non-mycorrhizal plants or non-mycorrhizal associated plants, couldn't do, right? So. And again, keep in mind that one was on canola ground, so I can say that the with the non-mycorrhizal inoculated plants, maybe they didn't have that same association. So very cool little studies. So really,
I assume they would eventually get the same association, or would they just have a way later start?
Yeah,
right?
Yeah. It's anyone's guess what's going on down there, but from what I see here, it's that August rainfall. So it's
but at that point, what I would say is the inoculated stuff, it had a fully functioning network from the very start. So when it didn't have much rain in August, it was prepared. Whereas the non-inoculated stuff, it would have to be natural spores find their way in and then propagate from there, which would just take time.
Yeah, especially because it was sown on canola ground, right? So
there just wouldn't be any there.
Yeah, it makes a heck of a lot of sense. There's no question. So we're gonna switch gears again, Joe. I think we're gonna try to keep this under an hour. So we got like 17 minutes here left. So, but cool. I wanted to make sure you touched on this, your full season cover crop.
Totally. This is my favorite crop in the world. I just heard a lot about full season covers, like the idea. This was a blend of, I think, 12 or 14 species. We were just trying to cover warm, cool season grasses, warm, cool season legumes, diverse brassicas, flowering plants, deep taproots, and as much mycorrhizal dependency as we could to mimic nature as best as possible. So We didn't use any fertilizer, no herbicide, no nothing. And the idea is to mob graze it as tightly as we can to put that diverse residue back on the soil surface. And the results were amazing. We did it again last year. We had very similar results, but I guess I'll touch on the mycorrhizal portion of this. So this is June 23rd, and you'll just see how quickly this crop is growing. So this is the 26th of June. You can see 3 days later, this was the first day we grazed. Just shot up like a like a weed. Yeah, so this is the 26th, first day of grazing. But the fact of the matter is, it was growing so fast. I didn't think I had enough animals there, so we started grazing them early. So this is, what is that, 20 days later? Yeah, our last day of rain was on the 9th of July. So it kind of gets even more impressive. You can see the cows laid down nice cover there. It's hopefully a balanced C to N. So that's going to break down really quickly. Feed biology and everything propagates, everything benefits. So we'll go to the last photo. Yeah, so this was 4 days later, the last paddock we grazed. You can see how tall it is. But the most impressive part, if you go to the next photo, Trav, is the regrowth. So that hasn't had rain in a month, and you know how tall sunflowers are in the start of August. It was unbelievably thick, and that can only be because of mycorrhizal fungi, water efficiency, and balancing that C to N.
Yeah, almost one of those things you got to see to believe. I'll touch on that a little bit later on, but that was my moment for sure when you showed me this.
Oh yeah, like I say, my favorite crop in the world. And then we went out and sowed fall rye with a disc drill into the residue, and we ended up grazing it the following spring again.
Yeah. Yeah, you touched on that. Just feed this to different soil microbes. So I know you sent me this just before, but can you explain this picture a little bit?
Yeah, so this is me putting up the wire on the left in the second last paddock. So that's the 16th of July. And then on the right is the 20th of July. And you can see that's the same quad in the picture. The one on the right, you can just see the tank at the spray tank on the top. But that's what we're talking about when like this. Who could you show that picture to and say that is no fertilizer? That is nature at work. Those plants have its symbiosis. There is a live fungal and network and biological network below ground, and there are no deficiencies there. And the growth is just so substantial and so rapid.
Yeah, truly amazing, Joe. That was, I guess, my aha moment. You know, seeing that was one thing, but actually benchmarking with the soil test for all you soil chemistry nerds out there, this is the soil that he grew that full season cover crop on. It defies everything we've learned in soil chemistry world. You know, how much biomass you created, you know, growing, you know, that's that diverse species mix, right? So, you know, we're looking at, you know, 8 parts per million phosphorus. We're looking at, you know, typically higher magnesium, lower like extremely low calcium. It's, yeah, it's just, it really did, you know, pull it all together for me to understand that, you know, when we start to utilize diversity, 1 + 1 doesn't equal 2 anymore. 1 + 1 = 3. And, you know, this picture depicts that probably perfectly.
Yeah, that picture kind of just gives an idea of what's going on below ground.
Yeah, just something else. All those plants, you know, mining nutrients from, you know, root depths that they've never seen before, right? And then sharing water with each other through the mycorrhizal fungal network, right? So
I'll say this too, Trav. Whenever I went out, I had a shovel with me because when you dug into the soil, you were just seeing soil like you'd never seen it before. I mean, yes, earthworms, but insects and aggregates and smell, like it was a pretty powerful experience, and obviously powerful enough that we're doing more and more and more, and I'm encouraging it as much as possible. But yeah, it's a pretty neat thing to do.
very, very, very cool, Joe. Definitely, that's a great summary as far as, you know, understanding the power of mycorrhizal fungi. But we're going to transition here. We're going to call this Fungi Talk. It's just a few slides that we wanted to throw together to chat about. You know, this one I actually got from Premier Tech yesterday. So what this is showing, you know, I see these fine filaments. This is actually your mycorrhizal hyphae network that's colonized the plant root. But what you see these I guess thicker lines, this is actually bacteria growing on the mycorrhizal hyphate network. So what we're seeing is the mycorrhizal fungi not only creating a symbiotic relationship with the plant, but we might be seeing a symbiotic relationship with bacteria as well here, which is truly, truly astonishing.
If I was a bacteria colony, I would want to set up next to a mycorrhizal fungal highway too, because it's a constant flow of nutrients.
Yeah, maybe that bacteria's response or their job is to go solubilize phosphorus, right? You can hitch a mycorrhizal fungi, that's benefit for both. So
they don't have the ability to transfer like the fungi does.
Yeah.
So it's pretty cool stuff.
Very cool. Yeah, incredibly cool stuff. So yeah, anyway, we'll move on from that. Again, just the microscope picture that I thought was pretty neat. We came across this one a little while ago as well, but this is actually a species of mycorrhizal fungi actually attacking a parasitic nematode. Obviously, it's under a high-powered microscope, but boy, is that fascinating stuff. If we can
that is amazing. Somebody got that on camera.
Yeah. So I don't know. We'll move on here from that. But again, just a few little tidbits of mycorrhizal fungi and how awesome the power is. So, Joe, you sent me, or I sent you this a long time ago. This was back when we were both kind of learning more about mycorrhizal fungi. But I don't know if you want to touch on this. I know it's one of your
totally.
Yeah.
Yeah, I would highly recommend people watch this. I'll try and keep it under a minute. But basically, this researcher went into the forest, and there were 4 different species of trees, 1, 2, 3, 4, all beside each other. So they hooked, put a tarp over one of the trees. Remember, there are 4 different species. Put a tarp over one, so the tree couldn't photosynthesize anymore, and they put traceable carbon into the veins of the 3 other trees. So put the traceable carbon in, covered the one species, and she went away and came back. And I forget the time exactly, Trav, but it was like 90 minutes. And within 90 minutes, parts of that traceable carbon were found inside the tree that was shaded just 90 minutes earlier. And from 3 different other species of trees. Like, what an amazing response time that was. And just, it shows just how, like, we can throw out this idea of competition. Like, it literally, when we talk about mycorrhizal fungi, which is what transferred that carbon, it's symbiotic. It's not competition. These plants are actually working with one another instead of against one another.
Yeah. I think that's another exciting part of research, especially in agriculture, is which plants are symbiotic with each other, which ones are can work together in this regard, right? So, intercropping, cover cropping, stuff like that. How can we look at building that network of symbiosis, right? So
totally.
But really cool. I'd highly, highly, highly recommend everyone hop on YouTube and pop that into YouTube. It changed my life. Joe, maybe you can say the same, but.
I think about it all the time. It is I think it's 20 minutes. It's excellent.
It was a great starting point to understanding mycorrhizal fungi. So we'll move on. I guess I always say too, if you don't believe us and all this mycorrhizal fungi talk, hop on Google, not just Google, but Google Scholar. It's just a compilation of research papers and academia research, but hop on there and you'll find over 100,000 published research papers on mycorrhizal fungi. So very fascinating stuff, very very cool stuff. So
YouTube, podcast, Trav, there's documentaries, there's lots of information out there, and trust me, you can't learn it all, but there's it's pretty cool to know, to understand the basics of it. But no, there's lots of information out there if you're excited or interested or excited to learn.
Cool stuff. Well, that's pretty much it, Joe. I do want to touch on our AGTIV product line. I know we've mixed it in a little bit here throughout the webinar. But yeah, here at Taurus, we represent AGTIV inoculant, as a lot of you know. That AGTIV inoculant not only has the Rhizobium bacteria, but has the mycorrhizal fungi in that granule or in that powder as well. So, you can talk to your local Taurus rep to learn more, or reach out to us on social media or whatever the platform might be. So, and I will strongly suggest Premier Tech and Taurus has done a lot of third-party research over the last 4 or 5 years. Highly recommend hopping online and downloading this research book. It is packed full of third-party research of our AGTIV product line. So hope you're surviving through this, Joe. But I know this is one of your favorite lines, but I've never let my schooling interfere with my education. It's a Mark Twain quote. So
that's a beauty.
Yeah, it is exactly kind of what we're talking about here. So different way to look at things, and how can we do things differently. So
Totally.
Anyway, Joe, that's it. I want to thank you for being here. That was fantastic.
Thanks for having me, Trav. Always fun talking to you.
Very good stuff. Well, on that note, we will wrap it up. Thanks everyone for listening.